Welcome back. All right, so we're moving on to chapter 7, and you'll see a lot of similarities with this one from the previous chapters. So this deals with meiosis primarily. Meiosis is how we're going to make your gametes, so your egg and your sperm. I spend a lot more time talking about mitosis because so you get those patterns that you're looking for, the different phases, and then you can use those patterns to easily identify them with meiosis, because you're going to see meiosis is just like mitosis, but there's a few changes in the Meiosis I. So let's get into it. So basically, meiosis is how we make the instruction manual for you. So if you remember, I've used the terms haploid and diploid, which let me go to that slide real quick and I'll come back. So haploid and diploid refer to terms how we can describe the number of chromosomes. So haploid means you only have one copy of every chromosome. This is going to be for your egg and sperm. Now, it's important that we only have one copy because, as shown right here, this is how we're going to make the process of going through fertilization. So we're going to have two haploid cells. We're going to have the sperm and the egg come together. So for humans, you have 23 chromosomes in your sperm, 23 in the egg. When these come together, we will have what's called your diploid cell. So if you remember, when diploid has both sets of the homologous pairs. So you have your maternal and your paternal copy. So you will have all 46. So two haploids, or two haps, come together to make the whole. So that's how we get all 46 chromosomes. So for each of these pairs right here, the sperm gave you one and the egg gave you one. Now, what's great about this leads to genetic variation. So this is called, another way is calling it sexual reproduction. Now, this is how we're going to get a combination of genes from both parents. So your parents gave you all that you have. So if you don't like it, blame them. So this makes up your instruction manual. Mom gave you 23 pages, Dad gave you 23 pages, and that's how we got to where we are. So that's a review on haploid diploid. So let's compare mitosis and meiosis. So mitosis is where we're making clones. What we have is we wind up getting two genetically identical cells. So the cells are exactly alike. It's like if we just put it in the copy machine and boom, got the same exact thing. Both cells that we make are going to wind up being diploid. All right, so we start diploid and we find out ending in diploid. This is how we make all your somatic cells. And we talked about, some organisms, this is how they go through reproduction, the prokaryotes, because they just wind up making clones of themselves. This is how we repair, replace, our growth. Key thing is chromosomes identical to the parental. So there's going to be no changes. We're making clones. Now with meiosis, our end product is 4 genetically unique cells. So they're going to be different combinations, and they're all going to be haploid. They're all going to have one copy. Now these are going to be your gametes, and the key thing is they are not exact copies of either parent. It's going to be a mixture of both. So I have it here on the top. Think of meiosis as mitosis twice. We're going to break it up into two. We're going to have what we call Meiosis I and Meiosis II. In Meiosis I, our goal is to separate the homologous chromosomes and reduce the chromosome number by half. This is where you're going to see a lot of the big differences. In Meiosis II, the goal is to separate the sisters. So this is just exactly like mitosis. So we're going to go spend a little bit more time on Meiosis I, and then Meiosis II, you should get and pick up pretty easily. So meiosis I, we're going to get prophase and prometaphase into the same slide right here. So what's happening is, same thing happening. The nuclear envelope is going to break down. Chromatin is going to condense into individual chromosomes. Nucleolus goes away. We're going to have those centrioles go and establish the poles. Microtubules are going to start to form. Now this is what's going to be different, is we're going to have the sisters are going to be paired together, and we're going to match them with the chromosomal homologs. So Reason we do this is we're going to do something called crossing over. And crossing over is where we kind of share parts of chromosomes with each other. And we want this to happen. This is not a bad time for chromosomes to switch part. So what happens is the homologous pairs join together. So this is chromosome 1. This is your paternal. We'll call this one your maternal. They join together to form this tetrad of 4. And this tetrad will hang out. And then they will go through, and what happens is they do this process of crossing over. So these internal chromatids right here will switch parts of each other, and they'll form what we call recombinant chromosomes. The external ones don't get to play with the games. Now, crossing over, it happens at specific points. There are hot spots where this happens quite often. We are able to measure that, figure out the frequency of crossing over. It's a lot of math. Wouldn't expect y'all to do that. But if you take genetics, it's actually kind of fun. But this is what's happening in prophase 1, crossing over. So the homologs line up together, so they'll form what's called that tetrad, and then they kind of share parts with each other. In prometaphase 1, the tetrads are going to be attached with the microtubule. So if I take my pencil right here, it's like that microtubule is going to go through and attach on both sides. So this is a whole unit, and it's going to stay together. Now in metaphase 1, we're going to have the tetrads, another term is a bivalent for them, are going to line up. So if you're in mitosis, everyone lined up like a single file line. But in metaphase 1, the tetrads stay together. So here's tetrad set 1, here's maybe tetrad set for chromosome 2. Now, something that happens that does lead to some genetic variation is called this random alignment. And this is one way that you get some genetic diversity. So this is a simple little organism, only has 2 chromosomes, so chromosome 1, chromosome 2. And what happens if the paternal all line up on the side, maternal all on the right side? So when we undergo meiosis 1 and they split up, you can see that this cell right here only got the paternal copies, and this one only got the maternal. And when we undergo meiosis 2, each of these, we'll call them little sperm. Like this sperm only got the parental copies, these only got the maternal. Now what happens if we just flip this middle one? So on the top one, paternal on this side, maternal, and on chromosome 2, we're going to flip it. So when we undergo meiosis 1, boom, boom, everyone is split up. Now this cell has a maternal and a paternal, but this cell has the opposite arrangement. And if you look, when we go through meiosis 2, the combination of the gametes. So this is how we get genetic variation. So just like in regular metaphase, everything's lining up in the middle. We're going to check to make sure the tetrads are going through the checkpoint. But big difference is the homologs are lined up, not just the sisters. The sisters are paired up with their homolog counterpoint. So next up is anaphase. So if you remember, an anaphase is where they split apart and they get pulled towards opposite poles. So what's going to happen is the homologs are going to break apart. Now, what's going to happen is this homologous pair is going to go one direction, and this is going to go the opposite direction. We're keeping the sisters together. Let's keep the sisters stay together. So we're just going to pull this set this way and this set this way. And what's going to happen in meiosis 2 is where we're going to break up the sisters. So the sisters are together in meiosis 1, and we split them in meiosis 2. So the big difference is we're pulling the homologs apart in meiosis 1. And then in telophase 1, we're just going to split it into 2. Envelope will be formed. Chromosomes can condense. Some of them enter what we call innerkinesis, a period of rest where they'll halt. If you want to think of it as like an interphase 2, but they do not replicate their DNA, they just hang out. Now, some cells, especially plant cells, they kind of skip this stage and they go straight into meiosis 2. So let me briefly explain the steps again with the picture images. So prophase 1 / prometaphase 1. Envelope goes away, nucleolus goes away, Poles are starting to get established. You know, microtubules are growing. Now the tetrads are going to get together and they're going to undergo what we call crossing over. So this homologous pairs, homologous pair, homologous pair. They're crossover sharing information. Metaphase 1, poles established, microtubules attached, boom, boom, boom. And now the homologous pairs are all lined up on this metaphase plate. Anaphase 1, split up the homologous pairs. The sisters stay together, so the sisters will still go towards their poles, but the homologs are broken apart. In telophase, envelope reforms, break down this complex, form the cleavage furrow, go through cytokinesis. Now we have 2 cells. Now meiosis II is just like mitosis, same exact thing. So you should recognize the patterns. In prophase 2, envelope breaks down, spindles start to form, you establish the poles, and this is going to happen to each of the cells that you made in this previous step. So now this is going to be from 1 cell to 2. Now in meiosis II, each of these cells is going to go through the same process. In metaphase 2, all the sisters line up here on the metaphase plate. In anaphase 2, sisters split apart. This is the big thing. We're breaking apart the sisters in anaphase 2. They stayed together in anaphase 1, and phase 2 is when we split them. And then telophase 2 and cytokinesis break down the complex. Envelope reforms, cleavage furrow, eventually form 2 cells. And then you will get 4 cells when you're done. Now each of these cells are going to be haploid. They're only going to have 1 copy of each chromosome. They're not going to have both the maternal and paternal. It's either or. So I like this image because it compares them side by side. So mitosis, we went from 1 cell to 2. Both of them are going to be diploid. They're going to have all copies of the chromosomes, maternal and paternal. We went through the patterns, and you can kind of recognize it. With meiosis, we went from 1 cell to 4 haploids. So you have a maternal, paternal, only 1 copy of each of them. Depending on the combination, how the chromosomes lined up will determine what you get. Meiosis I, the big difference are the homologs. The homologs joined together to form those tetrads. And that was when crossing over happened. They joined together, and then we broke up the tetrads in metaphase 1, anaphase 1, split them apart, and then we went for telophase. And then each of these cells went through meiosis II, which is exactly like mitosis. So they're a good figure to go and compare them side by side. So you get genetic variability in both ways. One way I talked about was an independent assortment, so how they lined up on that metaphase plate, you know, if all the maternal on one side, paternal on the other, if you flip just 1 pair. And the other way was crossing over. We have 23 pairs. That's quite fascinating. Think about all the combinations that you can have lined up on the metaphase plate. So you have, you know, 88.4 million possible combinations, even if there's no crossing over. So the combination that made you is 1 in 8.4 million. So it's quite fascinating, even though when you think about you have siblings, how similar you might look and everything. So it's a unique combination to make you. And then there's my dog at Muds, just because. So here's some review stuff for meiosis, and that wraps up this unit.